Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta, Emeritus Professor from Department of Biochemical Engineering and Biotechnology at Indian Institute of Technology, Delhi. Today we are going to talk on steroids from paper Structure and Function of Biomolecules 2. Steroids are structurally related and widely distributed compounds in animals and plants. Apart from sterols, which includes cholesterol, vitamin D, bile acids, adrenal cortex hormones, the so-called sex hormones, and sapogenins are all included in this list. At one time, a steroid was defined as any compound which gives diels hydrocarbon when distilled along with selenium at 360 degrees centigrade. So that is how our understanding about what steroids are developed. At higher temperature of 420 degrees centigrade, crysine and piscine are obtained. So the objective of this module are to learn about cholesterol structure, its precursors and its role in other biological structures, to learn about various sex hormones, to understand the role of corticoid hormones, and to learn about bile salts and their role in fat digestion particularly, and to understand how steroid hormones, what is their signal transduction mechanism, and that is by altering the gene expression. So the concept map is that the cholesterol molecule is biosynthesized from a simple acetate moiety, and cholesterol is the first important steroid molecule which is formed on this biosynthetic route, and it's from cholesterol that you get many of these derivatives, other steroids with other biological activities formed, like androgens, estrogens, corticoids, bile salts, and steroids. And those of these three, which are basically the hormones, steroid hormones, their signal transduction, we have to understand that how do these steroid hormones alter gene expression. That's overall concept. So let's step back and look at how our understanding about this class of molecules started. So one way of classifying lipids into two classes is that is saponifiable lipids and non-saponifiable lipids. So most of the lipids that we have talked about particularly so far are saponifiable. So saponifiable lipids are those lipids which upon hydrolysis by alkali gives rise to products which become water soluble. More strictly speaking, at least the products become more water soluble than the reactants. And this is the class of the lipids with which people were earlier familiar, and it was Cheverell who in 1812 observed that not all lipids are saponifiable. Colloid and Young in 1936 suggested to Royal Society of Chemistry, UK, that the compounds related to cholesterol should be called steroids, and that's where the word was used for the first time. Earlier, the word sterol existed for crystalline alcohols, derived from the Greek word of stereos, which actually means solid. So cholesterol was recognized as a C27 sterol. Sterols were known to be unsaponifiable or non-saponifiable lipids. Thus, cholesterol, the most widely known steroid, is actually responsible for this class of compounds named after it. The cholesterol structure determination involved some well-known scientists of that era: Wieland, Windans, Diels, Rosenheim, and King. The fundamental carbon skeleton in cholesterol is cyclopentane perhydrophenanthine. Quite a few steroids are synthesized by the mammals, and this includes cholesterol. In fact, in humans, cholesterol is the steroid which is present in largest amount. Human blood plasma contains total cholesterol of about 200 milligram per 100 ml. About 25% of this as free cholesterol. Almost all cholesterol, free and its esters, are present as lipoproteins. A standard test for cholesterol is Lieberman-Burkhardt reaction. In this test, acetic anhydride in chloroform solution is added to cholesterol sample in concentrated H2SO4 solution. Formation of a green color indicates presence of cholesterol. In fact, cholesterol and other sterols as well give this and few other tests. Addition of concentrated H2SO4 itself to cholesterol solution in chloroform turns the CHCl3 layer red. This is called Salkowski reaction. Cholesterol can be estimated in the form of its digitonide, which is formed when a saponin, digitonine's ethanolic solution, is added to its ethanolic solution. Cholesterol digitonide is a molecular complex with one molecule of each and forms white precipitate. It is curious that biosynthesis of this compound with a complex ring system is solely from a single source for its carbon atoms. That precursor is acetate. The isoprene is the early key intermediate in this pathway. In the 1940, Bloch fed isotopically labeled acetate and found that both carbons were part of cholesterol molecules in liver tissue slices. The incorporation of methyl group and carboxyl group carbons of acetate was treated in the synthesized cholesterol molecule and is indicated as M and C in this structure, which is shown M and C respectively. This was one of the early trumps of use of radioactive labels in determining biosynthetic as well as catabolic pathways of biologically important molecules. Before the simple precursors with radioactive labels were available, scientists did not have this approach available to them. Establishing the entire biosynthetic pathway involved extensive efforts by Bloch, Cornforth, Popcak, and Lyman. The importance of steroids was recognized by the industry very early. So, the group at Merck also made some key contributions. The determination of this pathway paved the way for many other important pathways, as much of the pathway is common to those pathways. The common perceptions about bad cholesterol and good cholesterol are based upon oversimplified communications about from the medical community. Cholesterol is same molecule in both cases. It's a case of being judged by the company one keeps. Cholesterol, like most of the lipids, moves between various tissues, organs in the bound form as part of lipoproteins. Lipoproteins are complexes of carrier apolipoproteins with cholesterol and its esters, phospholipids, and triglycerides. Low density lipoprotein LDL has highest percentage concentration of cholesterol plus cholesterol esters and is responsible for cholesterol associated with it to be termed as bad cholesterol. Higher systems avoid biosynthesis of complex molecules like cholesterol. Cholesterol is a very necessary molecule for several reasons. So, if enough cholesterol is available for its use in membranes, bile salts, steroid hormones, etc., activity of an enzyme HMG-CoA reductase is slowed down. The catalysis by HMG-CoA reductase is the rate limiting step in the biosynthetic pathway. Most of the cholesterol level lowering drugs are designed to inhibit the activity of HMG-CoA reductase. The intermediate HMG-CoA is also a common precursor for some other compounds. If cholesterol biosynthesis regulation is defective, serious consequences ensue. LDL is high and during transport it deposits cholesterol in blood vessels as atherosclerotic plaques. This causes difficulty in the blood flow and the condition is known as atherosclerosis. HDL, form of the bound cholesterol, represents good cholesterol. HDL, as it travels through in the blood, can pick up additional cholesterol. Atherosclerosis correlates positively with high levels of LDL and negatively with high levels of HDL in blood. Atherosclerosis leads to heart failure, as we commonly call it. The cause is obstruction of blood flow in coronary arteries. As the high level of HDL correlates negatively with atherosclerosis, that is why HDL bound cholesterol is considered a good form of the cholesterol. On the other hand, familial hypercholesteremia is a human genetic disorder associated with high levels of cholesterol in blood. It leads to severe atherosclerosis in childhood itself. The defect is in the LDL receptor, which mediates cholesterol uptake by the cells. As cholesterol is not taken up by the cells, the cholesterol synthesis continues. In this case also, inhibitors of HMG-CoA reductases like isovastatin, simvastatin, pravastatin and campectin are used in therapy. These drugs are commonly referred to as statins. Cholesterol was initially isolated from human gallstones, which mostly consist of this steroid. The natural sources of cholesterol are fish liver oil, brain and spinal cords of animals. Wool, essentially a protein, does contain some fat called lanolin. Lanolin is a mixture of palmitate, stearate and oleate esters of cholesterol. Sterols, the steroids is an alcohol group and the general class to which cholesterol belongs, occur in animals and plant oils and fats. During the early days, sterols obtained from the animal and plant sources were called zoosterols and phytosterols. If isolated from yeast and fungi, these were classified as mycosterols. Cholesterol, cholestanol and coprostenol were called zoosterols. Once the structure of steroids and their characterization was in place, it was realized that same compound could occur in many kingdoms. So this classification lost its meaning and is no longer used very widely. Sir Robinson and Woodward's synthesized cholesterol chemically. Apart from its complex structure, the challenge was the presence of asymmetric carbon atoms, which can give rise to 256 optical isomers. Bernal's work with X-ray diffraction showed steroid molecules to be flat. It also showed that hydrogen atom at C9 is trans to the group at C10. Some steroids have allo as a prefix in their names. Bernal's work established the way rings are fused in steroids. The compounds related to colistin are referred to as allo compounds. Those related to coprostin were called normal compounds. For the sake of simplicity, normal prefix is dropped and absence of any prefix means it's a normal steroid. It was Keller's group in 1936 which used optical rotation to characterize steroids. And it was found that all steroids with a double bond at 5 to 6 position are levorotatory. Cholesterol itself is a white crystalline compound with a melting point of 149 degrees centigrade and is levorotatory. It can be easily isolated from the yellow portion of the hen egg yolk. There's the hen egg yolk, the yellow portion of the hen egg. So many a times nuts like almonds and plant oils are described as cholesterol free. Plants, most of them don't actually contain cholesterol, although they do contain some of them in very small amount, or they contain cholesterol like molecules. So such advertisements are actually somewhat misleading. The plasma membranes on the other hand are rich in cholesterol. Cholesterol plays a major role in controlling the fluidity of these membranes. It increases fluidity by disturbing the regular array of fatty acyl chains in the membranes. At the same time, cholesterol prevents large movement of these chains. This yin and yang effect of cholesterol makes it very important molecule in eukaryotes. Hence, to perceive this as a bad molecule from health perspective sends very misleading message about its biochemical role. Cholesterol is well suited to be a compound of biomembranes as it is an amphipathic molecule. Brown and Goldstein, in their Nobel lecture in 1985, described cholesterol as the, quote, most highly decorated small molecule in biology. 13 Nobel Prizes have been awarded to scientists who devoted major parts of their career to cholesterol, unquote. Brown and Goldstein themselves were given Nobel Prize for establishing LDL pathway. The metabolism of dietary lipids present a challenge as lipids are insoluble in water-rich milieu of digestive tract of animals. The problem is solved by dispersing lipid in emulsions. Bile salts are more polar than cholesterol. This structural design makes these the designated amphipathic molecules for emulsifying the lipid molecules. Emulsions not only make lipids accessible to water-soluble enzymes, these also increase the surface area of the substrate lipids. This increased surface area also promotes intestinal absorption. Liver is the site of synthesis for bile salts. From there, these molecules reach gallbladder and are stored there. When needed, gallbladder releases bile salts in the small intestine. It is here that bile salts emulsify dietary lipids like fats and oils. Cholesterol itself is made in the liver, while a small percentage of it is incorporated as components of the membranes of hepatocytes. The other fats of cholesterol are expelled from the liver as biliary cholesterol, bile acids, and cholesterol esters. Thus, cholesterol is the precursor of bile acids. Choline-CoA is the intermediate in the synthesis of many bile salts. Glycocholate, the product of the reaction of choline-CoA with glycine, is the most important bile salt. Taurocholate, another bile salt, is the product of choline-CoA with taurine. Hormones are secretions of ductless glands. Pituitary hormones are polypeptides. So is well-known hormone insulin, which is secreted from the islets of Langerhans and pancreases. Adrenal hormones, adrenaline is a catecholamine. Gonads produce so-called sex hormones, which are steroid in nature. Their activity is controlled by some pituitary hormones produced by anterior lobe of that master gland. So these hormones are called primary sex hormones. The gonad hormones are called secondary sex hormones. These hormones are called sex hormones as these are involved in the sexual processes and the gender determination. The sex hormones are of three types. Androgens are male hormones. Estrogens are female or follicular hormones. Progesterone are the corpus luteum hormone. Cholesterol is the precursor of these and other steroid hormones. The steroid hormones are hence either adrenocortical hormones or sex hormones. These are synthesized in different endocrine tissues and reach the target cells through blood. The word endocrine is derived from two Greek words, endon, within, and krainin, that means to release. This distinguishes these from paracrine hormones and autocrine hormones. All steroid hormones have nuclear receptors. Binding of hormones to these receptors alters the gene expression. The word androgens is derived from the word andros for male. Two important steroid male hormones are testosterone and dihydrotestosterone. Dihydrotestosterone is formed in the prostatic tissue by reduction of testosterone by 5-alpha reductase. The other hormones with androgenic activity are adrenosterone, androstenedione 317 dione, 11 beta hydroxyandrostenedione 317 dione, dihydroepiandrosterone, and 17-alpha hydroxyprogesterone. Castration of young animals results in failure of the development of sex characteristics. In human males, castration prior to puberty inhibits ossification of long bones and reduces the ultimate height as well. Male sex hormones and female sex hormones have antagonistic physiological effects. The first androgens isolated with androsterone and dihydroisoandrosterone by Butant from male urine. Testosterone was first isolated in pure form from testes of bull. Cholesterol is converted to androgens in testes, ovary, adrenals, and placenta. Catabolic products of the androgens have been isolated both from the tissues and urine. Involvement of hormones in controlling the uterine cycle was known very early. Estrogens have been isolated from adrenal glands, testes, placenta and urine. Secretion of these hormones by the follicles of the ovary results in development of sex organs and other secondary gender characteristics like hair distribution and texture, skin texture, voice and fat distribution. In the absence of estrogens, human female menstrual cycle is disturbed. The estrogens are derived from androgens in liver and other steroid producing tissues. The estrogens and their catabolites are excreted in urine as conjugates of glucuronic acid and sulfuric acid. While natural estrogens are active and given parenterally, synthetic estrogen 17 alpha ethanyl estradiol and stibestrol can be given orally. Androgens are precursors of estrogens, estrone and estradiol, which are formed by aromatization of a ring and loss of a methyl group. These enzymatic reactions depend upon both NADPH and oxygen. As we saw, the androgens in turn are synthesized from progesterone. In fact, cholesterol is first converted to pregnenolone. Pregnenolone in turn produces progesterone. Cholesterol, a C20 steroid, loses a C6 side chain to form pregnenolone. All these reactions which are shown are again dependent upon NADPH and oxygen. Pregnenolone is the precursor of all steroid hormones, so its formation is a critical step in steroid biochemistry. So this is regulated by ACTH, which is also called corticotrophin. ACTH is synthesized by the anterior pituitary gland. The conversion of pregnenolone to progesterone is a two-step process. First is the oxidation of 3-hydroxyl to a 3-keto group. The second is the shifting of the double bond from 5 positions to the 3rd position. Progesterone is also the precursor of cortisol and aldosterone, which is a major glucocorticoid. These conversions require enzyme catalyzed hydroxylation reactions. Conversion to aldosterone also requires oxidation of C18 methyl group to the aldehyde group. The deficiency of 21-hydroxylase involved in the synthesis of glucocorticoids and mineralocorticoids constitutes most common inherited disorder. High levels of androgens results in accelerated growth and short stature in males. In females, masculinization of external genital organs may result. The adrenal cortex produces corticoid steroid hormones. These are called mineralocorticoids or glucocorticoids. Production of glucocorticoids is controlled by ACTH, a hormone of anterior pituitary. Decreased production of glucocorticoid due to deficiency of 21-hydroxylase makes ACTH secretion go up via feedback loop. High ACTH level increases conversion of cholesterol to pregnenolone. Aldosterone's primary sphere of influence is electrolyte and water metabolism. Cortisol administration results in increased glycogenesis and gluconeogenesis from amino acids. To a varying degree, adrenal steroids influence multiple biochemical processes such as carbohydrate, protein and lipid metabolism and even inflammatory and allergic processes. About 30% of humans deficient in 21-hydroxylase lose sodium ion in urine continuously. The adrenal steroids regulate the concentration of sodium and potassium ions in extracellular fluids. Aldosterone and deoxycorticosterone are known to result in increased reabsorption of sodium chloride and bicarbonate ions in the kidney distal tubules, sweat glands, salivary glands and gastrointestinal mucosa. Excess hormonal concentration causes rise in extracellular sodium ion concentration, an expansion of extracellular volume and decline in potassium ion concentration in serum, elevation of blood pressure and deleterious effects on cardiac muscle activity. are the clinical manifestations. Signal transduction mechanisms of hormone follow diverse mechanisms. This is understandable in view of their diverse structure and physiological functions. Many hormones bind to cell surface receptor to initiate signal transduction. Steroid hormones, on the other hand, directly affect gene expression, hence their target is cell nucleus. A critical difference between steroid hormone effect is that while other hormones produce results in minutes, steroid hormones take hours to reach full impact. This delay effect is due to the time taken in gearing up the protein synthesis as a result of change in gene expression pattern. The sequence of events is that a steroid hormone binds to a receptor, the receptor steroid complex enters the cell nucleus, the complex binds to the DNA and then alters the gene expression. Affinity chromatography with steroid as affinity ligand facilitates purification of hormone receptors such as those corresponding to estrogens, progesterone and glucocorticoids. Antibodies against such receptors also constitute valuable screening probes for the proteins expressed by library of cDNA clones. Such studies have revealed that receptors contain DNA binding sequences which are rich in cysteine, arginine and lysine. So receptors act as homing devices to take steroids to the gene. The steroid binding regions of the gene are in proximity to the DNA binding region. Steroid binding promotes transcription. Steroid hormones are not the only ones which operate in this fashion. Even thyroxine also forms a complex with the receptor, and the complex acts as a transcriptional enhancer. In that system, the insight came from the fact that VERB-A oncogene of avian erythroblastosis virus binds ERB-A gene in the host cell. This gene also happens to be the gene for thyroxine. In both cases, the receptors contain DNA binding regions which have sequences typical of metal binding fingers. Such metal binding finger domains are known to exist in wide variety of both DNA binding and RNA binding proteins. Such domains may have had an ancestral gene from where these domains were incorporated in nucleic acid binding proteins. Slide 24. The specific DNA regions to which the hormone receptor complexes bind are called hormone response elements. Hormone binding to the receptor itself induces a conformational change in the receptor. This allows receptors to interact with the transcriptional factors. HREs for various hormone receptor complexes are similar in length, but their nucleotide sequences vary with each hormone. This consensus sequence is thus specific to each hormone. Each sequence has two sequences, either together or separated by a nucleotide trimer. The hormone receptor complex binds as a dimer, each monomer's zinc fingers binding to nucleotide 6 sequences. Apart from HRE sequences, its number and position related to the gene also varies with each hormone. Slide 25. Many plants are rich in steroid glycosides, the so-called neutral saponins. The aglycones of these are called sapogenins. Some other steroid glycosides found along with saponins have effect on cardiac activity. Used in adequate amounts, these compounds stop heart function. Aborigines used to apply these on the tip of their arrows to kill enemies. The important aglycones of these glycosides are digitoxygenine, periplogenine and saramatogenine. Used in appropriate amounts, these cardiac glycosides can stimulate mammalian heart activity and hence have medicinal value. The venom of some toads are also steroid derivatives, and an important toad steroid is gamma bufetylene. In these venoms, the steroid forms esters with subryl arginine. Steroids find large amount of applications which includes their therapeutic action. Steroid transformations are an active area of biocatalysis. Microbial cells in free and whole cell forms have been extensively used in such steroid transformations. The specific DNA regions to which the hormone receptor complexes bind are called hormone response elements. Hormone binding to the receptor itself induces a conformational change in the receptor. This allows receptors to interact with the transcriptional factors. HREs for various hormone receptor complexes are similar in length, but their nucleotide sequences vary with each hormone. This consensus sequence is thus specific to each hormone. Each sequence has two sequences, either together or separated by a nucleotide trimer. The hormone receptor complex binds as a dimer, each monomer's zinc fingers binding to nucleotide 6 sequences. Apart from HRE sequences, its number and position relative to the gene also varies with each hormone. Many plants are rich in steroid glycosides, the so-called neutral saponins. The aglycones of these are called sapogenins. Some other steroid glycosides found along with saponins have effect on cardiac activity. Used in adequate amounts, these compounds stop heart function. Aborigines used to apply these on the tip of their arrows to kill enemies. The important aglycones of these glycosides are digitoxygenine, periplogenine, and saramatogenine. Used in appropriate amounts, these cardiac glycosides can stimulate mammalian heart activity and hence have medicinal value. The venom of some toads are also steroid derivatives. An important toad steroid is gamma bufetylene. In these venoms, the steroid forms esters with subryl arginine. Steroids find large amount of applications which includes their therapeutic action. Steroid transformations are an active area of biocatalysis. Microbial cells in free and whole cell forms have been extensively used in such steroid transformations. To summarize, in this module we have learned that cholesterol is an important and essential constituent of biomembranes. All steroid hormones and bile salts are produced from cholesterol. Androgen and estrogens are sex hormones. Adrenal hormones are glucocorticoids. Steroid hormones act by altering gene expression. We have discussed steroid hormones in this module. A better appreciation of this hormonal action is possible by integrating the knowledge about all hormones. There is a lot of connectivity to be discovered that way among various hormones and even within steroid hormones. A case in point is that we exemplified cortisone as a glucocorticoid. It doesn't merely affect carbohydrate metabolism, but nitrogen and even fat metabolism as well. Furthermore, deoxycortisone also is involved in electrolyte balance, so it functions like a mineralocorticoid as well. Finally, steroid glycosides are fascinating molecules producing diverse physiological actions. Thank you.